In the last lecture, we finished right here, where we talked about the fate of proteins that are being translated on an mRNA. What happens to them? Like those that remain in the cytosol, those that go through the secretory pathway or display on the plasma membrane, or those that go to other intracellular organelle. Now, in this lecture, we will start the process of uncovering some of the molecular players. So before we get there, so this is just the figure from your textbook where the same experiment that I explained earlier is elaborated. That the proteins that are inserted into the endoplasmic reticulum, either because they are destined for the cell membrane or because they are destined for the secretory pathway, then those proteins are protected from a protease digestion until and unless the vesicles are treated with detergent, so which demonstrates that the proteins are protected within a lipid membrane vesicle, in the lumen of a lipid membrane. Okay. Now, the question that everyone was in everyone's mind is that why is it that only non-cytosolic proteins should be directed to the ER? Okay, like proteins that are like on the mRNA, ribosomes are translating, and then some of the proteins that are destined to stay in the cytosol. So they are not directed to the ER. Why the other proteins are directed to the ER? Now, here in this lecture, we will primarily deal with that aspect. So you know the mechanism. So what we are going to explain here is the process of investigation that uncovered the mechanism, right? So you know that there is a signal peptide that it is recognized by something. Okay. Now, recognition by something is fine, but then it has to be inserted inside the membrane. So there has to be a receptor on the membrane. And then you know that the signal peptide is cleaved. So there has to be a peptidase that will cleave the membrane. So all these different molecular players, how they were discovered is what will be the subject of this lecture. Okay. So, as I said, that Sabatini and Palladie knew that the large ribosomal subunit is instrumental for attaching the ribosomes to the ER membrane. However, they could not molecularly define the interactions between the ER membrane and the ribosomal subunits. So there enters Gunter Blobel. Gunter Blobel is a mentee of Palladie. They received their Nobel prizes just 25 years apart. And Blobel began just where Palladie left, with the aim of molecularly defining the process. Palladie pretty much described exactly what is happening at a phenomenal level, but Blobel entered and uncovered the major molecular players in the recognition of the nascent peptides that are destined for secretory pathway, and the molecular players involved in that. So in the meantime, both Palladie and Blobel and others showed that there is no inherent difference between the ribosomes. The free and ER-bound ribosomes are interchangeable, and they are the same ribosomes. So there is nothing special, like one might have thought that the ribosomes that are translating secreted proteins are a different kind of ribosome, and ribosomes that are translating soluble proteins, the different kind of ribosome. So, and that is perhaps underlying the difference, but that turned out not to be the case. So then came the famous signal hypothesis, that you all know about. I am reading from Blobel's own writing. We postulated that all mRNAs for secretory proteins code for a signatory amino terminal sequence element that is recognized by a soluble factor that in turn binds the nascent chain ribosome complex to the ER membrane. And then he actually drew a diagram in 1971. You can see that this is a hand-drawn diagram. And like there he talks about that there are these ribosomal subunit pools, the small subunit, the large subunit. This is the mRNA 5' to 3'. The small subunit first comes, then the large subunit joins, and then the protein is synthesized. This small asterisk is what he referred to as the signal peptide, which will be bound by something which he called the binding factor here, the binding factor. And then this binding factor will bind only to those proteins that have this, and then it will tether it to the membrane, and then it will be vectorially injected inside the membrane, and then the protein synthesis will end, and the small and large ribosomal subunits will come apart, and then they will become part of the pool, and this will keep on happening. Please note the year 1971. At that time, there was no evidence for any of this. And I was fortunate that I attended a lecture by Gunter Blobel himself, where he stated that nobody in the world believed his hypothesis. Every grant that he would write to investigate this hypothesis would be turned down. They will not be funded. So he will write all sorts of mundane grants for mundane projects and save some money aside to chase his dream, this like testing this hypothesis, and the rest is history. But you need to appreciate the power of logic and conviction that Blobel had. So Blobel thought through all the evidences beforehand that if the ribosomes are not different, if only the secreted proteins are being targeted to the ER, and then they are being released, so there must be something that can recognize only the secretory proteins. And there was already evidence with IgG, etc., that there is a special sequence in the terminus which is not there in the mature protein. So he connected all the dots. So this is what is a hallmark of a fantastic scientist. That is, you connect the dots, come up and filling the missing links with solid logic-driven hypothesis, and then you test it. Either you prove or disprove your hypothesis. Either way, you will make discoveries which will contribute to the literature significantly. So then let us see how he went about proving his hypothesis. So he was a very, very good biochemist. Okay, so what he did is that he used a chemical called puromycin, which kind of looks like a tRNA. So puromycin will be mentioned multiple times in this lecture. So it is better that you understand what puromycin does. So puromycin mimics structurally a tRNA. So it comes and sits at the A site, and then the peptide chain that is at the P site will be transferred onto puromycin, and this puromycin peptide chain actually just slips through the ribosomal tunnel, and the elongation is stopped. Okay, so puromycin is a translation elongation inhibitor, and high salt is very often used by biochemists to dislodge interactions. Interactions are often mediated by either ionic interactions or hydrogen bonding interactions. Both can be disrupted with high salt. So this is a very common technique by good biochemists to disrupt non-covalent interaction between biomolecules, mostly proteins and protein with something else. So using these two, he could free up the microsomal membranes from ribosome. So you remember microsomal membranes are fragments of the ER. So he can free them up. And he could purify pure, large, and small ribosomal subunits. He could isolate pure mRNA. Of course, there is no molecular biology in those days, okay? Like no recombinant DNA technology. So he cannot make his own mRNA. He will use cell types where a particular mRNA is likely to be produced in a very high amount. Like if you go for red blood cells, you can, or erythrocytes, you can get beta globin, which is produced in very high amount. So that's how he will do. Like he will get pure mRNA, but does not mean pure mRNA for a particular protein. It's pure mRNA for the whole cell, which will predominantly have one species or the other. Okay, so if he takes this, then if he simply in vitro synthesizes the protein, okay, so you have the mRNA, you simply add S35 labeled methionine, which will be incorporated in the protein. All the machinery needed for protein synthesis, like we used to call it translation extract, you add it, and then the radioactive protein that will be made will be directed by this mRNA. And because you are having a certified methionine, so it will be radioactive. So if you do that, then you get this product, which is called the pre-light chain. Now we know that it is pre-light chain that is slightly bigger in size. If you purify the pre-light chain that is secreted by myeloma cell, which is like blood cell type, then you see that protein, this is a naturally processed and secreted peptide protein, right? That protein has slightly smaller molecular weight than the in vitro synthesized protein. So a part of it is lost. But if you do in vitro synthesis, you do not get the smaller size. Now, if you do that, if you look here, line number 3, in vitro readout of polysomes prepared by detergent from isolated rough microsomes of myeloma cell, meaning you have all of this and you provide microsome, okay? Like you do it in the presence of the rough microsome. Then you get the larger one, which you get anyway from in vitro synthesis, and you get the smaller one, which you call the mature light chain. So everything taken together, that showed that like the IgG light chain contains an extension of amino acid in its N-terminus, 2 to 3 kilodalton, which is not present in the mature peptide. And it was only IgG synthesized in presence of microsome that produced the shorter form. Okay, so therefore the microsomal membrane will contain a protease that processes the nascent IgG into the mature one, right? So that was the first hint that there is something in the microsomal membrane. Now he makes 3 major postulates. That the signal peptidase, which chops the protein. Okay. The signal peptidase must be membrane associated. It must be on the luminal side of the ER, meaning it cuts the protein on the, like if this is the ER membrane, it cuts it on this side, on the luminal side, and not on the cytosolic side. And the 5' ribosomes in a polysome will have signal peptide containing proteins, while 3' associated ribosomes in a polysome will have proteins without signal peptide. So this is a very, very critical statement that is being made here. What is actually being said, what it actually means is that the processing actually happens co-translationally. That is, initially it is produced, the signal peptide, it is there as part of the protein, and even before translation is finished, it is chopped off. So the 5' ribosomes in a polysome will have the signal peptide, the 3' ribosomes will not have it. Now see how the experiment is designed. It is not, like you are not used to looking at such experiments. So let us say that this is the mRNA, okay? This is 5', this is 3'. And these are all the ribosomes that are, crawling over it, okay? Now, they all have proteins coming out, like here it is rather long, here it is relatively short, and here it is like this, here it is like this, a bit different length. Now, this is being done in the presence of microsome, okay? In vitro synthesis of protein, but in the presence of microsome, the time course of readout of detached polysomes, okay? In a cell-free translation system. So first, len1 is the major in vitro translation product. Okay, so that gives you the size of the pre-light chain, and then this is the secreted one from myeloma cell that gives you the size of the mature light chain. Now they're adding S35 methionine and then stopping the reaction after 1 minute, 6 minutes, 9 minutes, or 18 minutes. Now think what happens. You have added the S35 methionine, and then after 1 minute, which proteins will be complete? The synthesis will be complete. These ones, right? These ones. So one, it will incorporate very little, like, let me see if I can. Okay, so first of all, it will incorporate very little length, and so it will be very faint. So you see here, it is very faint. So it is very faint, and it is of the same size as the matured light chain. Ones that will complete the protein synthesis after 6 minutes will be somewhere over here or here, okay? Like these ones, so they will finish the thing. So here they will have opportunity to incorporate a little bit more radioactivity. Here it could incorporate only for 1 minute. Here they will be able to incorporate for 6 minutes. Therefore, the signal level, the radioactivity incorporated will go up, but they are still the smaller sized. Once that will incorporate it for 9 minutes, then perhaps it will be like this one. Okay, so it will incorporate for more longer time. So yes, even more stronger signal, but now you see a faint band here, which becomes prominent at the 18th minute. Like there at the 18th minute, you are perhaps looking at this ribosome. So this ribosome, when the ribosome was here, at that time it had the signal peptide, okay, and it got into the like it could be cleaved, and you also have the matured light chain, the pre-light chain and matured light chain. So therefore the ones that you were getting, like the ribosomes that are at the 5' end of the mRNA, they still have the extra 2 to 3 kilodalton signal peptide, but the ones that are at the 3' end, they do not have that extra one. So what it means is that as the ribosome is crawling through the mRNA and the secreted protein is being synthesized, somewhere along the line between right after the starting and right before finishing, somewhere that 2 to 3 kilodalton signal peptide is getting chopped off. Okay, and because it happens only in the presence of the microsomal membrane, therefore the signal peptidase must be membrane associated. And yeah, and later experiment will prove that that it is on the luminal side of the ER. Of course, there are some papers that I have recommended here. You may want to read at it, look at it. So here it is, another way of looking at it to demonstrate that the presence of microsome is necessary to generate the small form. That is, if you just do the in vitro transcription, you get the larger form. You do the in vitro transcription in presence of a protease. Of course, there is nothing to protect that protein, so it will be completely degraded. You do the in vitro transcription in presence of microsome, you get the smaller form, and as well as the larger form, which you get with just in vitro transcription. You do it in presence of microsome and protease. You see that the smaller form is not at all disturbed, but it is the larger form which is gone. Because what is before it gets into the microsome, it is accessible to the protease. Therefore, this experiment proves two things. One, that the presence of the microsome is necessary to generate the small form, and the small form is protected from the action of proteases. Therefore, the peptidase must be on the trans side of the luminal side of the ER, while the larger form is not protected because that is yet to enter the the microsome, the luminal space, so the protease can access it. And they also did some centrifugation studies and demonstrated that the mature chain co-sedimented with the vesicles. Okay, so fine. So now we know that this is real. Okay, that the secreted protein, as it is being synthesized, it gets inserted into the ER membrane, in the ER lumen. And then eventually they will be encapsulated within small vesicles, and they will be resistant to proteases if you just add protease. But if you first destroy the lipid bilayer, then they will be accessible to the protease and they will be degraded. Okay, and also they showed that the translation and translocation occur simultaneously. That is, if you first do the in vitro synthesis and then add the microsomal membrane, then if you add protease, the proteins will be degraded. So microsomal membrane, if you provide after the proteins are already made, then they cannot get into the luminal side. That's why they are getting degraded by the protease, right? On the other hand, if you have the microsomal membranes present while the proteins are being synthesized in the cell-free in vitro system, then they are protected from the action of proteases, because they are then inside the microsomal vesicle in the luminal side. So it shows that the translation of the protein and translocation into the ER must happen simultaneously. If the translation is first made, I'm like you know that if you do the translation in vitro, the larger form is met, right? So therefore the signal peptide will be there. But even if the signal peptide is there, it will still not be able to get inside the vesicle. So therefore, the there has to be an involvement of the ribosomes in the process of them getting inside the ER. Like, because like the mature proteins, like the proteins whose translation has completed, they are no longer associated with the ribosome, so they cannot get in. Only the translating only those proteins can get in which are in the process of getting translated. So ribosomes must play a role in the translocation and peptidase activity and all the paraphernalia. So therefore Blobel proposed a new theory, okay, in 1975. Like, this is a little faint, it is from his Nobel lecture. So he proposed that there will be a channel, okay, which will be a translocation channel. The ribosome, as it is scanning the mRNA and synthesizing the protein, will sit on that channel and will insert the protein inside the channel, and that's how the whole thing happens. So this was, he proposed in 1975, okay, which could not be proven till 1991. So this is how insightful a scientist was Gunter Blobel, okay. Now, the moment you invoke a channel protein, it will necessarily require a way to have the proteins inserted into the membranes. Okay, so then he went on to prove the entire mechanism leading up to synthesis of the protein, being recognized, getting tethered to the channel, and then getting inside the ER lumen. Okay, so then he thought that the channel should be some kind of like then the question was that what do how do the integral membrane proteins get inserted in the membrane. So his postulation was that he postulated model for catalyzed integration into the lipid bilayer of integral membrane protein. These are all models, 1975. Okay, so what he said is that they will get inside the channel, and then there will be something that will trigger opening of the channel within the bilayer of the lipid membrane. And once it opens, the protein will simply move out into the bilayer. And now we know that that is true. So there is something called a ER translocation signal, which is the signal peptide. Then there will be a ER translocation stop transfer signal. When that stop transfer signal is hit, that's when the channel would open laterally. Laterally meaning on its side within the plane of the lipid bilayer, and the protein will move out. So this is from your textbook. So the co-translationally, the protein gets inserted into the channel, and then there is a signal peptidase that cuts it, and then the channel, when the stop transfer patch hits the channel opens laterally, and the protein moves out. So this is happening because the membrane is fluid mosaic, so proteins can migrate within the plane of the membrane, and all it requires is that the channel to open up. And for the channel to open up, you need the stop transfer signal. So that was his postulation, and now we know that that is true. Now he moves on to to further demonstrate that what happens to the transmembrane protein. That the transmembrane protein will have certain part that is exposed to the outside, not on the luminal side, but on the cytosolic side of the vesicle, and they will therefore be accessible to the protease. So how would you do that? Simple. So this time he is using VSVG protein. And VSVG protein, if you just translate them in vitro, you will get the full length protein. Okay, so this is the transmembrane segment. This is, now we know that this is the part that hangs outside of the cell, and then there is this signal peptide. But because it is being translated in vitro, there will be no sugar modification. Okay. You do that in presence of protease, of course, there is nothing to protect it, so most of it is degraded. you do that in presence of microsomes, then you get a slightly higher molecular weight form, which will not have the signal peptide, but which will have the this sugar modification. Okay, and of course you have the one with the signal peptide removed as well. And now, in the presence of the protease, you get a slightly smaller size. So unlike IgG light chain, in presence of protease, the size is not changed. Here, the size changes slightly, okay, from here to here. Why? Because this part hangs outside of the microsomal membrane. This is the cytosolic side of the ER membrane. This protein is hanging out. So there it gets chopped off. Okay? So he demonstrated that. Now comes the issue of how the mRNA or the protein that is being synthesized, we can even call it nascent protein in the process of being synthesized. How does it get to the ER? Okay, so there has to be something that recognizes the signal, which is called the signal recognition particle. If you listen to Blobel's talk, you will see the reference of a brilliant graduate student called Peter Walter. Peter Walter is now a very, very celebrated scientist at University of California, San Francisco. He's one of the doyens of cell biology. So what Peter Walter did, in the hindsight today, it might sound very simple, but if you listen to the lecture, you will realize that it required a lot of blood and sweat to do the thing. So if you take microsome and salt wash it, that is, you will strip it off all the membrane associated things, including the ribosome, and any you will know, right, that there are integral membrane proteins and there are peripheral membrane proteins. Peripheral membrane proteins are associated with the membrane but not necessarily inserted within the membrane. Okay, so those will also be washed off with high salt. So salt washed microsome, there was no translocation. So I should explain it to you a little bit. So this is the Eppendorf tube. You have the microsome. Now you add very high salt containing buffer, mix it very well, spin it. Once you spin it, then it gets into a tight pellet, and then you have this liquid. You take it out in another tube, and this is your salt wash fraction, and you have the membrane separately. Okay, so you now add that salt washed microsome and the salt wash fraction, this one, and then you see translocation. So there is something that was associated with the membrane peripherally, loosely, which is required for translocation, and that is present in this thing. So this is purely a liquid, right? Like a buffer suspension. So therefore, there is something in here that allows the translocation to happen. And we already discussed all sorts of assays to determine translocation. So then you simply fractionate this salt wash fraction, you do all sorts of chromatography, and eventually Peter Walter purified a 6 subunit protein complex, and he called it the signal recognition particle, or SRP. SRP was quite surprising that among other things, it also has a small RNA as a 7s RNA component. So this is the structure of the SRP, and they drew this cartoon that this is the small subunit, the large subunit, the protein comes out. This is the signal peptide. As soon as it comes out of the ribosomal tunnel, then SRP recognizes it, and then it recognizes it. Okay, so this SRP is recognized, required for translocation. So now the structural work has been done, and a lot more insight is gained about the function of SRP, which I am not particularly knowledgeable about. Now, so the peptide that is making protein that is destined to make a secreted protein or a plasma membrane protein is recognized by the SRP. Fine, that part is fine. Now, how is it that because you saw that is happening in the absence of the membrane, right? So because it is in the salt wash fraction. So now how will this be recognized on the membrane of the endoplasmic reticulum? So there has to be a receptor for the SRP. How would you identify that one? So next, the assay is to purify the SRP receptor. Okay. So here, if you listen to the lecture or read the paper, I do not recall exactly where I saw this. They had a very, very frustrating time. Okay? So they tried all sorts of sources for SRP and for the ribosomes, and they finally could hit the perfect combo. That is, they will take canine pancreas SRP, meaning SRP purified from the dog pancreas, and they will take ribosome from wheat germ. Okay. Don't ask me that why this one works and not any of the other combo. There are some of these strange things happen in biochemistry. Most often people do not chase the causes for this, but we know that this happens. So to cut a long story short, the Blobel's lab, they established that the canine SRP, signal recognition particle, has low affinity towards wheat germ ribosomes. But if the canine, the wheat germ ribosomes are bound to mRNA encoding secreted proteins, then canine SRP has high affinity towards it. But if it is translating mRNA that are encoding soluble cytosolic protein, then there is very low affinity. So, that fits the hypothesis, right? That if the ribosome is translating a secreted protein, then that will be recognized by the SRP. If it is not translating a secreted protein, then it will not be recognized by the SRP. Now, when SRP interacts with the ribosome, there is a translational arrest. Okay, that is very, very important. There is no membrane now. Okay, all you have are mRNAs on which ribosomes are calling, and there is protein coming out. If there is SRP, then SRP will bind to it and there will be a translation arrest. And the translation arrest happens the protein will be around, I think, 7 kilodalton or something like that. You will soon see it. I don't remember. So interaction of SRP with ribosomes did not cause translation arrest if it was translating a soluble protein. But if it was translating a secreted protein, then there will be a translation arrest. And it is around 70 amino acids. So 70 amino acid is around 7 kD. Okay. So the length of the arrested nascent protein will be 70 amino acid. This translation arrest, again, please understand that this is happening in the absence of any ER membrane. This arrest will be released if you add microsomal membrane. Okay, so that means that there is something on the microsomal membrane which is releasing this translation arrest. And then, they postulated that there must be a receptor on the microsomal membrane, interaction with which is releasing the arrest. So then they, purified that receptor from the microsomal membrane, and the assay this time was that what is there from the solubilized microsomal membrane which can release the translation arrest in here. Okay, so again, try to understand that this arrest is without the membrane. With the membrane, arrest is released. So to identify the receptor, they took the membrane, solubilized it, and then fractionated it. These are very, very different techniques, very difficult techniques. Okay, if you talk to Professor Arun Shukla, you will realize that dealing with membrane proteins is extremely difficult, because membrane proteins are, generally speaking, not soluble in water. Okay, because they love to live in a lipid environment, so they are generally hydrophobic protein. So extremely difficult, technically extremely difficult proposition that we are talking about, but they did it. So they solubilized the microsomal membrane, fractionated it, and identified a fraction which, if added, the translation arrest is released. So that's how they identified the SRP receptor. So now you got two of the components. Okay, signal peptide was already shown. Then they got the SRP receptor, SRP and SRP receptor. Okay, so again, like the structural data, and I am not good at it. Now, what removes the signal peptide? So there will be a signal peptidase. So how did they purify that one? So signal peptidase again will be present in the solubilized membrane, and that can accurately chop the protein. Okay, so you can have the in vitro synthesized system, and you add solubilized microsomal membrane and see if it is getting processed. And if you biochemistry is like the approach of biochemistry is just like genetics. In genetics, the operating principle is that there must be a gene that is doing a particular function. So you do not know which gene it is. You turn off every gene, one gene at a time, and see which one, like which mutant, cannot perform the function. So you say that, okay, so then this gene must be important for this function. In biochemistry, you identify a sample, okay? So a specimen. In this case, it is the microsomal membrane, so solubilized microsomal membrane. So that has the ability to chop the signal peptide. So solubilized microsomal membrane is therefore your superset, okay? And this superset, you fractionate it by different chromatographic techniques into, like 1, 2, 3, many fractions. And then you identify one fraction wherein it still has this ability to chop the nascent peptide. And that's the way you purify eventually the protein. We call purification of the activity, then identification of the protein. So using this activity, chopping the nascent peptide, they purified the peptidase, and they figured out that it is the peptidase has 5 non-identical protein subunits, and all of them are integral membrane proteins. Okay, so this is the actual purification gel. This is the lane 1 will be the solubilized membrane. So you can see so many proteins are there, and then different purification steps eventually to arrive at this very pure protein, which is the peptidase. You can see 1, 2, 3, 4, 5 bands. Okay, so it's a 5 subunit, 5 non-identical subunit peptidase, and they had other assays to demonstrate that all of these are integral membrane proteins. Okay? So let me stop this lecture right here. In the next lecture, I will talk about a very, very exciting story of how did they demonstrate presence of the protein conducting channel, this thing. Yeah, this. How did they demonstrate the presence of a protein conducting channel? So that will be in the next lecture. Okay. So